Yeah, my table of contents include introduction to herbal medicines and quality control, standardization and quality assessment, regulatory frameworks, innovations in quality control. As you all are aware that, sorry, herbal medicines are the formulations containing one or more plant parts or plant extracts possessing certain therapeutic values which are used to treat various ailments and improve overall health. Herbal medicines have a rich history that spans thousands of years. The World Health Organization estimates that approximately 80% of world's population relies on herbal or traditional medicine for their primary health care. Now coming to the quality control. Quality control is a part of good manufacturing practice. So way back in our pharmaceutical formulations, there was a drug called as thalidomide, and that was prescribed for the pregnant women to prevent morning sickness. And this drug, when it was launched into the market, it created a tragedy that is still called as thalidomide tragedy, where the babies born were with deformed upper limbs and lower limbs. And till date, those victims are existing in Europe. And such many incidents happened in many parts of the world because of not taking proper care of manufacturing processes. Then onwards, they implemented good manufacturing practices where they brought the component called as quality control. The quality of each and every product which is launched into the market has to be thoroughly checked for its purity, safety and efficacy for the well-being of the end user. So, importance of quality control in herbal medicines. The regulatory framework for herbal products is not as rigorous as synthetic pharmaceuticals, resulting in an inadequate quality control measures. As I just mentioned, the regulatory authorities are very much particular about synthetic drugs. They don't even allow a single molecule to enter into the market with lot many stringent procedures. For a single drug to launch into the market, it may take more than 10 years, because under these the duration of 10 years or more than that, the molecule is tested for its safety and efficacy, then only it will be launched into the market. There are various regulatory agencies throughout the world like US FDA, EMEA, and our Indian regulatory agencies CDSCO. These are the authorities which control the manufacturing, sales and distribution of the pharmaceutical product. But when we look into the regulatory authorities looking into the pharmaceutical herbal drug products are not that much stringent, and that is the reason most of the time when they are consumed by the patients, lot many adverse effects are seen. So all the other countries now, they have brought lot many guidance or guidelines into the system where they can look into that the product which is consumed by the patient is quite safe and effective. This lack of oversight can create significant safety hazards for consumers, as herbal medications may contain contaminants, adulterants or improper formulations. What is the reason for such adverse effects is most probably that is due to the presence of contaminants or not proper usage of right herbal product, that is called as adulterant. Sometimes they mix up with similarly looking herbs into the formulation to reduce the cost of the product, or because of various reasons. When the actual drug is very expensive, they try to adulterate with the same looking herb, and which will not be effective, or sometimes it may cause some harmful effects also. By implementing stringent testing and quality control measures, potential risks such as contamination, adulteration and presence of harmful substances can be minimized, ensuring the product is quite safe for usage. Coming to the key elements of quality control, safety and efficacy. Quality control is vital to ascertain that herbal medicines are safe for consumption and exhibit effective therapeutic results by ensuring rigorous testing and evaluation. Potential contaminants, toxins and variation in potency can be identified and controlled, thereby safeguarding the consumer. That is the main aim of the quality control, to cater safe and effective medicines to the society. and consistency in therapeutic applications. So when we look into the consistency of the therapeutic applications of the same herb, that may vary because of various reasons, that is variability in herbal formulations, that is again due to variation in the cultivation as well as collection and environmental methods. All these can lead to variability in the effectiveness of the product. Now coming exactly into the main part of the quality control, that is quality control tests for herbal medicines. These are the quality control tests which are mandatory to be followed or to be maintained in the pharmaceutical industries, and each and every document pertaining to these things have to be documented. In our pharmaceutical industries, we have a process called as auditing. There are different types of auditing. Among that, the stringent auditing is the regulatory auditing, where when they come for inspection or auditing, they ask each and every document which is to be maintained by the pharmaceutical industry, and that is a rigorous process, and wherein they need to maintain the document for each and everything. And the quality control are divided into 3 stages. One is quality control of raw materials, and next is in-process quality control test, which is called as IPQC, and third one is finished product quality control test. So what are the quality control parameters that are subjected to raw materials? One is macroscopic evaluation, which is also called as organoleptic properties. Here the crude drug, when it is supplied by the vendor, it is being subjected for its purity test, identification test, and based on various organoleptic properties, sometimes if the person is expert, they can easily identify the product which is being supplied by the vendor is the same or different one. For example, Ashoka bark. Okay? Ashoka bark is a crude drug which has got many therapeutic values, and sometimes that is being adulterated with similar looking bark, because that is to be collected from the forest, that is not cultivated. And these types of herbs which are mostly collected from the forest, they are more prone for adulteration because they are difficult to collect from the forest, and they have very vital therapeutic values and all. That's the reason they will be adulterated with similar looking drug products or crude drugs, and with proper identification the evaluation can be done. That is called as macroscopic evaluation. Some are having particular texture when it is break up, that should show the particular texture on the breaking part of the bark or root or whatever it is, then that indicates that is the identity of that particular crude drug. And looking in, going through the color as well as the smell of the crude drug, one can easily identify whether that is the same or it is adulterated one. Like this, the quality control is qualitatively evaluated by macroscopic method. And next is microscopic evaluation. Microscopic evaluation is mainly done for the powdered drugs. Okay? When the drug is supplied in the form of powder, that cannot be easily identified through observation through the naked eyes, because that is a complex mixture. And for example, if a particular product, assume it is a Nux vomica powder. When the Nux vomica powder is need to be identified, it is observed under microscope after proper staining. So when you can see the lignin cells around in the microscope, then you can get the confirmation that it contains, it is a product of Nux vomica or it is the powder of Nux vomica. Similarly, by observing into certain cells and tissues, we can get the confirmation about that particular crude drug. Then foreign matter. So many of the time when the drugs are collected from the forest, especially the roots and other ground level parts of the plants, they get mixed with sand and other materials which are not to be present in the particular crude drug. Again, that need to be purified before subjecting for further processing. If it is not subjected for purification, then the same foreign matter will remain in the product and spoils the end product. That's why the foreign product need to be thoroughly cleaned, and the pure one need to be, sometimes the same plant part which we need in a particular form, we are interested only in the floral part. and sometimes it may contain other part like leaves and birds and other things that need to be clearly excluded from the main floral part, which is the main part of the plant which contain active ingredient, and further it is subjected for processing. Next is ash value. Hope all of you might be knowing all these things. Some of you are botanists, and some of you use these types of evaluation parameters in your studies also. But as a part of this quality control study, I am briefing about this. Ash value is nothing but it is again a qualitative measure to identify the crude drug, because each and every drug, when it is subjected for ash, the percentage, based on the percentage of ash what is obtained compared to the actual weight of the drug, that denotes the type of crude drug, or that gives in the identifying a particular type of crude drug. Based on that we can also identify, not confirmatory, these are not confirmatory tests, okay, but still we can come to know whether it is that particular drug or it is something else. For example, if a pepper is having around ash value of 8%, means then if it is showing after converting the pepper, the crude drug is heated into ash, and that ash weight is recorded and it is compared with the actual weight, and based on the percentage of ash obtained, we can get the qualitative identification of the product. If the pepper value is 8%, the ash value of pepper value is 8%, and there is a deviation, either it is more or less, then we can come to the conclusion that it might be not pepper, or it might be adulterated with some other material. Next is microbial contaminants and aflatoxins, and this evaluation, that is microbial contamination, mainly happened during storage, when the medicine is containing more than the specified quantity of moisture content. It can allow the microorganisms to grow, because water is the source for the multiplication of microorganism, and when the limit of moisture content is beyond the specified extent or specified limit, then the chances of microbial growth will be there, and that can spoil the product when it is subjected for processing. In the initial stage it may look safe, but when that product is used for herbal formulation manufacturing, and the end product will be completely spoiled during storage. When it is used by the consumer, or before it is used by the consumer, it will show its degradation, microbial degradation. And similarly, these microbials may be either E. coli or Salmonella species, fungi, and other types of microorganisms that can release their metabolites, end products, when they are developed and present in the drug products. These may be very harmful. Amongst that, aflatoxin is one of the toxic substance which is produced by the fungi, which is one of the very harmful chemical substance which is highly carcinogenic in nature. And these types of materials can be identified either by microbial assay. Okay? The product is subjected for microbial assay by using different types of culture media, bacterial culture media, fungi culture media, and aflatoxins can be determined by HPLC technique or LC-MS technique. And aflatoxins, there is a percentage for microbial contamination or limit for microbial contamination, but there is no limit for aflatoxin. It should be completely free from aflatoxin. Any of the drug product or herbal formulation, the guidelines says that it must be completely free from aflatoxin, because it is highly carcinogenic in nature, and lot many work nowadays, lot many research is going on to identify the presence of aflatoxin. Lot many analytical methods have been developed, lot many analytical equipments are coming into market where they are helpful in detecting these aflatoxins. Next is solvent and pesticide residues. Most of the time we use different types of solvents to extract the active principle, exact active principle, to isolate the exact active principle. For example, a particular active principle is soluble in petroleum ether, then we make use of petroleum ether. When we extract it, we get only that particular component as a isolate, and when we subject it for drying by using either distillation method or by using steam, another type of process, any drying process, either by tray drying or spray drying process, sometimes chances are there the solvent will remain as the residue in the herbal product, which may be harmful when that is consumed by the patient or the user. And such solvents can be identified by various chemical method. If seen, that raw material can be avoided for further processing, which is subjected for again, or which will be sent back to the vendor, because many of these things are supplied by the vendors. No pharma industry is directly involved in isolating the extracts or growing the medicinal plants and all. These things are procured by the vendors. So again, the material will be rejected and sent back to the vendor if any such residual solvents is present. And coming to the pesticides residue, these are the herbal preparations which are cultivated in various locations for estimation and all. They will ask only the HPLC data because it is the most reliable analytical technique. And column chromatography, it is mainly used for isolation of various compounds from a single drug product. For example, an extract is prepared, and this extract contains many complex structures, and each and every complex structure need to be separated by using column chromatography. Based on the affinity to the stationary phase, the molecule which is having most affinity to the stationary phase will get separated first. Likewise, based on the affinity, the molecules get segregated, and further they can be collected separately by using particular solvents. Next is high performance, HPTLC actually. Before that, I read it as HPLC. HPTLC is again the sophisticated version of the thin layer chromatography. Okay, there it is done by using instrument, whereas thin layer chromatography is done manually, and HPTLC is done by using HPTLC instrument, where the fragmentation or fingerprinting can be done easily. Then gas chromatography or mass spectrometry, GCMS, is mainly employed for the separation of volatile substances like edible oils, as well as not edible oils, volatile oils, and many other type of volatile substances can be easily fragmented by gas chromatography. If a volatile oil contains different types of molecules, then those can be separated by using GCMS. And even some of the biological samples are subjected for this is most commonly used by again for the identification of any toxic substances or poisonous substances in biological fluids. Next is spectroscopic techniques, that is UV and IR. It is again used as a quality control tool to identify the presence or absence of a particular compound. Okay, by UV spectroscopy we can come to know if the reference standard is there, then the sample is compared with that, if that is matching with the reference standard, and roughly we can come to the conclusion that is the same compound. And IR is mainly used to know the nature of the molecule, that is whether they are proteins or whether they are alkaloids or saponins or flavonoids. So by looking into the functional group, of the molecule, we can easily identify the molecule, whether it is a particular alkaloid or a flavonoid, whatever it is. Yeah, these are the studies which are done in our research. Yeah, with HPLC chromatography, that is curcumin standard curves, and this is the sample which is developed or extract which is extracted having the same RT. Yeah, this is again one more which is compared with the marketed product, and this is the toxic substance which is found in ginkgo biloba, as well as pesticide and aflatoxin, which is determined by liquid chromatography. Yeah, again regulatory frameworks need lot many collaborations with World Health Organization, USP, European Pharma, and AISH. Yeah, due to time constraint, I am just concluding my session with the last slide. Yeah, integration is good practice. Good agricultural and selection practices, that is GACP guidelines, focus on best practices in the cultivation and harvesting of medicinal plants. Following these practices helps minimize variability in raw materials, contributing to the overall quality of herbal medicines. GMP Adhering to GMP ensures that products are consistently produced and controlled according to quality standards. This regulatory framework addresses all aspects of production, including hygiene, environment, and equipment standards. Thank you.